Hello my dear students, welcome back to the another amazing biology class on our channel. Today we are going to uncover the secrets hidden inside every tiny seed, that is seed structure and germination. Have you ever wondered how a small dry seed wakes up and grows into a big green plant? What parts does a seed have inside it, and what changes happen when it starts to sprout? Stay with me till the end, because by the time we finish this lesson you will understand how seeds protect new life and how they burst into beautiful plants when the conditions are just right. Let's dive in and discover the magic of seeds together. So our syllabus includes fruit and seed definition and significance, structure of dicot and monocot seeds, germination of seeds and types and conditions, structure and germination of bean seed and maize grain, differences between monocot and dicot seeds, hypogeal and epigeal germination explained and supported by experiments. What is a seed? Before learning how seeds grow, you should know the meaning of fruit, seed and grain. Fruit is a large ripened ovary, the part of the flower where seeds develop. The fruit protects the seed and helps it spread to new places. Examples: mango, pea pod, etc. Seed is a ripened ovule. It has a tiny baby plant inside called an embryo. The outer covering or seed coat protects the embryo from damage. Examples: bean seed, peas, etc. This is the image of the seed. This is the image of a fruit. This is the seed inside. This is the fruit wall. And grain found in crops like maize and wheat. It is a fruit where the fruit wall and seed coat stick together to form a tough protective layer. More about seeds. After fertilization, a seed forms after fertilization when male and female cells join. Contains embryo. The seed holds a tiny living embryo which develops from the egg and sperm cells. Dormant stage: the embryo stays inactive or dormant until good conditions help it grow. Food material: seeds have stored food to feed the embryo while it starts growing. Survival: the seed coat protects the embryo if conditions are bad, too cold, dry, etc. Some seeds can stay dormant for many years, even up to 100 years. Structure of seed, that is a dicot seed or monocot seed. It has endosperm, it has embryo, endosperm, epicotyle, hypocotyle, radical, cotyledon and seed coat. Diagram of seed germination: radical, seed coat. These are the cotyledon, first leaf, epicotyle, cotyledon, hypocotyle, root, etc. Classification and structure of seeds. Types of seeds. Seeds are basically two main types: monocotyledonous or monocot seeds, one seed leaf or cotyledon, example maize, grasses, etc. Dicotyledonous or dicots, two seed leaves or cotyledons, example pea, gram, bean, etc. Seed vary in size. Some seeds are very small like poppy or orchid seeds. Some seeds are quite large, example watermelon or mango stones. The largest seeds are coconut and double coconut. These are the seeds of orchid showing the image. This is the mango seed, and these are very small seeds, and these are the coconut seed which are very large in size. So seed structure, though size and shape vary, the basic structure of seeds are similar in nature. On the basis of endosperm, albuminous seed, these are also called as endospermic seed. They have endosperm for food storage that stays. Dicot albuminous, poppy and custard apple. Monocot albuminous cereals, millets, palms, etc. X albuminous seed are non-endospermic seed. No endosperm when matured. The cotyledons store food and become thick. Dicot X albuminous, it has gram, pea, mango, mustard, etc. Monocot X albuminous, the Vallisneria, orchids, Amorphophallus, etc. These are the diagram of endospermic seed or albuminous seed, and these are non-endospermic seed or X albuminous seed. The bean seed. There are many types of beans like broad bean, kidney bean, French bean, etc. They usually have a kidney shape and a convex surface. Seed coat: it has testa or hard outer covering. It protects the seed from damage, bacteria, fungi and insects. Tegmen is a thin layer under the testa, adding extra protection. Hilum, a scar on the seed where it was attached to the fruit or ovary wall. Micropyle, a tiny pore near the hilum. It lets water enter the seed for germination. also helps in exchanging gases for the embryo. Role of micropyle, it soaks up water for the embryo to grow. It allows gases to pass in and out for the baby plant. Cotyledons, two thick structures that store food for the growing baby plant. Embryo, it is found attached to one cotyledon. It has two parts, radicle which grows into the root and plumule which grows into the shoot with tiny leaves and a growing point. Maize grain. What is a maize grain? It's a seed where the fruit wall and seed coat are fused. This makes it a grain. Features: one side has a light spot showing where the embryo is. Most of the grain is endosperm, rich in starch, food for the embryo. The endosperm and embryo are separated by a thin layer called as epithelium. The outer endosperm layer is called the aleuron layer, which is rich in protein. Embryo of maize, it has only one cotyledon called the scutellum. It has a radicle or root and a plumule or shoot. Protective sheath, coleorhiza, it protects the radicle. Coleoptile, it protects the plumule. Maize grain, quick fact. The maize grain is a monocot structure and endospermic. It has a large endosperm for food storage. Other examples include rice, wheat, oat, etc. Major differences between bean seed and maize grain. Bean seed has two cotyledons. Maize has one cotyledon which is called as scutellum. Bean has no endosperm, but maize has large endosperm. Bean has a large embryo, but maize has smaller embryo. In beans, plumule leaves folded. In maize, plumule leaves are rolled. In beans, plumule are large. In maize, plumule leaves are rolled. Plumule large. In maize, plumules are very small. In beans, hilum and microbiles are visible. In maize, hilum and microbiles are not visible. In beans, seeds are separate in a fruit or pod. Maize, seed wall and fruit wall fused. A single grain structure is formed. Germination. A seed holds a dormant embryo which is held in resting or inactive state. It looks lifeless, but its life activities are very slow inside. Dry seeds breathe out carbon dioxide and little heat. When good conditions are given, the embryo wakes up, becomes active and starts to grow. This is called germination. The process of a seed turning into a seedling or young plant. Process of seed germination. This is the image showing the process of seed germination. Conditions necessary for germination: water. Seeds take water mainly from soil through the micropyle. Water is needed to swell the seed, break the coat, help roots come out, start chemical reactions that help move to the baby plant, help food move to the baby plant. Suitable temperature: very low or very high temperature stop germination. Base range 25 degree Celsius to 35 degree Celsius is the optimum temperature for seed germination. Tropical seeds need higher temperature than seeds of temperate areas. Oxygen. Its seeds need oxygen for respiration, energy making process. Respiration gives energy for the embryo cells to divide and grow. Seeds planted too deep may not grow because not enough oxygen reaches them. Weak pushing force, the baby shoot or plumule cannot push through the thick soil. Experiments on germination. It proves that water is needed for germination. Take two beakers. Beaker A, wet cotton wool and seeds. Beaker B, it has a dry cotton wool and seeds. Keep them in the same conditions. In beaker A, seeds will germinate, seeds will germinate because there is a wet cotton wool and seeds, and from the wet cotton wool the seeds will get the water. But in beaker B, seeds will not grow, proving water is essential. Idea is that without water, seeds stay in dormant condition but can't turn into plants. This is the image showing need of water for the germination of seed. Experiment to prove that a suitable temperature is necessary for germination. Take two beakers, name them A and B, put them wet cotton wool in each beaker and place some green gram seeds on it. Keep beaker A in a normal warm room and keep beaker B in a refrigerator in cold condition. In 1 to 2 days, seeds in beaker A, which is present in normal room temperature, it will sprout, showing that a suitable warm temperature is needed for germination. But seeds in beaker B may not germinate at all or may take a very long time, showing that very cold temperatures slow down or it can stop germination. So the image showing experiment need of temperature for the germination of seeds. Experiment to prove that air or oxygen is necessary for germination. Take two conical flasks, name them A and B. Spread wet cotton wool in both flasks and place soaked gram seeds on it. Have a small test tube inside flask A with alkaline pyrogallic acid which can absorb oxygen. Make sure that there is no chemical touches the seeds so that it can be harmed. Fix the test tube with a cork at the top of the flask. In flask B there is a test tube with plain water. Keeps both flasks in a warm place. Result: seeds in flask B germinate, showing oxygen is needed, but seeds in flask A do not germinate because there is no oxygen. Maybe very tiny growth happens due to little oxygen. So this image proves that oxygen is necessary for seed germination. 3 bean experiments. Tie 3 soaked bean seeds to a glass slide in 3 different positions. Top seeds are only in air. Middle seed touches both air and water. Bottom seed completely under water. Keep the slide in a beaker with water, so top seed is above water, middle seed is half in water, bottom seed is fully immersed in water. Result: middle seed germinates, it has both air and water. Top seed does not germinate, gets only air, no water. Bottom seeds does only germinate or stops after tiny roots comes out, gets only water and very little air. This proves water and oxygen are both needed for germination. Types of germination. Important parts: epicotyle, the stem region above the cotyledons; hypocotyle, the stem region below the cotyledons. If the epicotyle grows, hypogeal germination, cotyledons stay underground. Example: pea and gram. If the hypocotyle grows, in case of epigeal germination, cotyledons come above ground. Example: castor, bean, etc. In case of hypogeal germination, cotyledons stay underground, but in case of epigeal germination, cotyledons come above the ground. In hypogeal germination, epicotyle grows, but in epigeal germination, hypocotyle grows. Method to observe stages of germination: take a glass vessel, jar or beaker, fill it with a sterilized sand, clean and germ free. Line the inner wall with filter paper or ordinary white paper. Place seeds between the paper and the glass wall. Add water to keep the sand moist, not flooded. Seeds absorb water from the moist paper and sand. After a few days, you can observe seed swells, testa or seed coat breaks, radicle or root grows downward, and plumule or shoot grows upward. So the benefit is easy way to watch germination step by step without disturbing the seeds in the soil. Germination in some common seeds. Pea seed by hypogeal germination. The pea seed takes in water and it becomes swells. The outer cover or testa softens and bursts open. The radicle or baby root grows downward to make the root system. The plumule or baby shoot grows upward to make the shoot system. In early stages, the plumule bends into an arch to protect itself while coming out of the soil. The cotyledons or seed leaves stay underground. They supply food to the seedling until it can make its own food. This type of germination is called hypogeal. Hypo means below, and geo means earth. Bean seed epigeal germination. The bean seeds soak water and swells. The radicle grows downward to form roots. The hypocotyle grows and forms a loop or arch that pushes above the soil. This brings the cotyledons above the soil. Cotyledons act like first green leaves and help make food for the young plant. This type of epigeal germination. This is the image showing the mode of epigeal germination of bean. Maize grain, which is undergo hypogeal germination. The maize grain also takes in water and swells. The radicle pushes through its protective cover or coleorhiza and grows downward to make roots. The first root dies soon, but new roots grow. The plumule grows upward through its sheath or coleoptile to form the shoot. The cotyledon or scutellum stays underground and absorbs food from the endosperm. The hypocotyle, what is the region below the cotyledon, does not elongate, so cotyledons stay underground. So the germination is hypogeal. Viviparous germination, it's a special case. It is found in mangrove plants like Rhizophora and Schoneraceae. Seeds start germinating while still attached to the parent plant. The seedling develops a root while still on the tree or twig. When mature, the young seedling falls into a muddy soil and grows straightway. This helps mangrove plants survive in swampy, wet places. This is viviparity, twig of Rhizophora, showing viviparous germination, seedling growing in mud. Viviparity, it's a special case. It means that seeds germinating inside the fruit while it is still attached to the parent plant. Example, mangrove plants like Rhizophora and Schoneraceae. After the seedling grows enough, it drops into the soil and fixes itself by growing roots. The seedling germination ends with the formation of a seedling. A seedling is a young plant that grows from a seed and starts to live independently. Its roots absorb water and minerals from the soil. Its leaves make food by photosynthesis to help the plant grow bigger and mature. The mature plant then produces flowers and seeds, starting the life cycle again. This is the points to remember. A dicot seed has an embryo with two cotyledons inside a seed coat. The embryo has a radicle, that is baby root, and a plumule or baby shoot. Cotyledons store food for the embryo. A monocot seed has one cotyledon, large endosperm, and a small embryo with a small plumule with rolled leaves. Cotyledons conditions for germinations include water, suitable temperature, oxygen. Germination can be hypogeal. In that case, cotyledons stay underground, and in the case of epigeal, cotyledons are pushed above the soil. So dear students, today we learned how a tiny seed hides so much life inside it with its protective seed coat, nourishing food supply, and the baby plant called the embryo. We also saw how when conditions are right, the seed wakes up, absorbs water, and begins the wonderful journey of germination to grow into a new plant. Is not it amazing how nature packs so much power into something so small? Thank you so much for watching this. If this video helped understand seed structure and germination better, please don't forget to like, share and subscribe to the channel. Keep learning, stay curious, and I will see you in the next class with more fascinating lessons from biology. Take care and happy learning.